This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbiotix, this is TWiV, This Week in Virology, a special episode recorded on December 18th, 2018. I'm Vincent and you are listening to the podcast all about viruses. Today we are recording in Worcester, Massachusetts. We are at Microbiotix, a small biotech company here in Massachusetts, and I have 4 members of the company to talk to me about what it is that they do and maybe why. And joining me today at the end of the table, if you're watching the video, and there is video for this episode, the CEO of the company, Terry Boland. Welcome to TWiV.
Hello, welcome to Microbiotix.
Microbiotix, so I said it right.
You did.
All right, thank you.
Well done.
On my left, we have the Director of Chemistry, Zach Aaron.
Yep, that's correct.
Aaron, not Aaron, right?
No.
Welcome to TWiV.
Thank you.
Do we have good chemistry here? We'll find out.
I hope so.
Great chemistry.
On the other side, actually, I don't know you in Islam. You can tell me what your position is. Grace Liu, welcome.
Yes, thank you.
So what's your title?
I'm a senior scientist here.
A senior scientist, SENIR-SCIENTIS. Do you always wanted to be a scientist? You are now.
Yes, I am, and I'm glad to be here.
Thank you, Grace, for coming. And are you also a senior scientist?
And the new biosafety officer.
Oh, that's
How'd that happen?
Last but not least, Islam Hussein, senior scientist and biosafety officer. Welcome to TWiV.
My pleasure.
Your first TWiV, right?
It's my first, but I wrote in several times.
Although you have interviewed me and I have interviewed you before.
Yes.
And we are no strangers to each other. It's actually you that have arranged all of this.
My pleasure, yes.
This trip to Massachusetts, yesterday at Tufts, today at Microbiotix, and this is where you work, and we're going to hear more about it. So let's start with each of you telling me a little bit of your history. I'd love to know where you're from, where you were educated, and all that. Let's start with you, Zach. Where are you from?
So I'm originally from the Chicago suburbs in Illinois. I did my undergraduate work in chemistry at University of Illinois in Champaign-Urbana. I then went to University of California at Irvine, where I did my PhD with Professor Overman, studying the total synthesis of complex alkaloid natural products. After that, I went to Boston, where I worked with Chris Walsh on the enzymological biosynthesis of natural products. From the Walsh Lab, I actually started an independent career at Indiana University in the chemistry department as an assistant professor, where I was for, I believe, 6 years, working on biomimetic synthetic methodology, as well as some novel fluorescent imaging agents. I left Indiana in 2013. I went to Celgene for about 18 months, where I did covalent medicinal chemistry, and then I came here. And I've been here for approaching 3.5 years now, I think. I'm not very good at math.
So from your years at Harvard, you must have known Jim Hogle, right? You and Chris Walsh collaborated sometime. He did the polio structure years ago.
So I think that was before the time I was with Chris that was happening.
Okay. He's still there.
But the active collaboration, I mean.
Okay, got it. All right, Terry, what's your history? Where are you from?
Ohio. I went to Ohio State. Still a season ticket holder.
Well, you never get rid of them, right?
Never. Then I went to Cleveland State for graduate school. And I did all my graduate research in the immunology department at the Cleveland Clinic. It was a murine retrovirus lab. However, most of my research was tumor immunology, looking at T-cell responses to fibrosarcoma and the role of cytokines. The cytokines were lymphocyte activating factor, T-cell growth factor, which were renamed later as interleukin-1 and interleukin-2, so that slightly dates me. Then I went to NIH, joined yet another murine retrovirus lab. A guy named Jim Eiley had just discovered interleukin-3. So I did some of the early characterization of interleukin-3. I came up with a monoclonal antibody, and we used that in a lot of the early characterization. And that path got me a job at Dow Chemical. So I joined the Dow Chemical pharmaceutical division. They needed a tumor immunologist, basically. And I joined the oncology group as a tumor immunologist. And the Merrill Dow research, which I was part of, the way they did the research was very biochemical pathway directed. So sort of the therapeutic component was second to the pathway. And I worked a lot on the polyamine biosynthetic pathway initially. And eventually, by the time I left there, I was director of immunology. And then in the mid-90s, I moved to Montreal and joined a Canadian pharmaceutical company called Biochem Pharma. I was head of research. It was mostly oncology and antiviral drug discovery and development. I helped to set up Microbiotix while I was there in the late 90s. And then I joined Microbiotix full-time in 2002. And I've been the CEO here doing anti-infective small molecule drug discovery since 2003.
Okay. And you still live in Ohio, right?
I still have a home there, and I've had homes various places around the world, but I still have one there.
Okay. Grace, where are you from?
I'm from China. That's actually where I got my bachelor's degree in polymer science and engineering. And then I came to the US for grad school, and that's actually it's at Clark University in Worcester, maybe a few miles from here. And I got my PhD in biochemistry, working with Dr. Noel Lazo on a small peptide called IAPP. It's islet amyloid polypeptide, which is associated with the death of beta cells, which secrete insulin. So we believe it's associated with type 2 diabetes. And after that, I went to Washington University working with Dr. Gaia Emerasinger and Dr. Daisy Liang on structural biology on viral proteins from Ebola virus. And in 2016, I started working here, continue virology work.
So you're relatively new hire here.
Yep.
Okay.
I'm the newest.
Islam, you're the newest. So tell us your history. I know you're from Egypt.
I'm from Egypt. That's where I got my DVM degree. And soon after I graduated, I was hired as a teaching assistant in the same institution where I graduated from. And you just have to climb up the ladder until you become a professor. But I left that at some point, and I did a master's degree. So I was hired as a teaching assistant at the virology department. We had a separate department for virology, which is not very common in Egypt. So I did a master's degree on chicken anemia virus, a small circovirus. And then I was very lucky to get a scholarship to do a PhD in Cambridge University, UK. So I moved to the UK and did a PhD on antivirals, antiherpetics, with Hugh Field and Lawrence Stiley. And after I finished my PhD, I moved to the US and I was a postdoctoral fellow at the NIH in the National Cancer Institute in Frederick. And I did some basic research on HIV for about 3 years. And then I moved to Kansas, to the medical school in Kansas University, where I worked on hantavirus for about 2 years. Got some nice experience on working under BSL-3 conditions. And then I was also lucky again to move to Massachusetts, where I got a job at MIT as a scientist. And I worked with Ron Steyler Lab, where we did a TWiV yesterday. And I worked as on influenza viruses. And last year, I was lucky one more time to come here and work for Microbiotix. And I work on several antiviral projects.
Okay. And we're going to talk about what you each do. But first, I'd like to hear a little bit about the history of the company from you, Terry, from way back. You said you helped start it. So
Yeah, so the company was incorporated in 1998, pretty late in 98. And the founders, two of which are from UMass across the street. They were working on bacterial polymerases. They identified some uracil derivatives as potential antibacterials. And they spent most of 1999 trying to find funding for a new company they were setting up called Microbiotix. And they visited our labs at that time in Montreal. And we had a lot of nucleoside work going on at that time, including a lot of polymerase work. So I thought it fit pretty well with some of our ongoing research. And the company, Biochem Pharma, decided to invest in their new company, Microbiotix. And the labs officially opened in this building, I believe, January 2nd, 2000. So we're rapidly coming up on our 20th anniversary in this building. So we've been around a while. And as you may know, biotech companies age in dog years, okay, right? I mean, so it's 20 years is a good run. And the focus of the company is small molecule anti-infective drug discovery and development, covering a pretty wide range. So the initial focus of the company was exclusively antibacterial. We moved into antivirals. We now have a beta gamma herpes inhibitor that's now ready for phase 2 human studies, and a number of other programs that I'm hoping you'll hear about from our colleagues here. We actually have one malaria project, which is quite interesting, and we just got a small grant on our first fungal project. All of these projects that have been carried out here, without exception, we have numerous academic collaborators and government collaborators. So the model here is a little bit unique, I think, to the biotech industry, that not only do we have a core group of scientists here who are involved in discovery and development, we have literally dozens of collaborators that we work with, have gotten funded with, to advance our anti-infective programs.
So you're not supported by venture capital investors?
Yeah, we've never had any VC money. We've had some corporate money, including our seed money we had was corporate. I want to acknowledge and thank, though, that we've had NIH support. We've had Department of DoD support. We now have CARB-X support. So we've had a number of non-dilutive sources for our research.
Are there other small companies that use that model of mainly running on grant money?
A few, but not many. There's certainly plenty of other companies that have gotten started with that money. Then they transition to other types of funding. Yeah. And we also may transition, now that we have a clinical program, we may also transition to some corporate or venture capital funding.
Yeah, I think after we talk about the project, I'll come back and ask about your future. But you were in an academic lab in Montreal, right?
No, I had an academic appointment at the University of Montreal. I've had several academic appointments, but I was full-time with the company. So I was head of research at Biochem Pharma, which was a big research group.
So your entire career was in industry? My
entire primary appointments have always been in industry, but I've also had a number of adjunct appointments as well.
Okay. So Islam, let's talk a little bit about what you do here.
Well, I work on several projects. Primarily, I'm assisting with the clinical development of a compound called MBX400, which is we are trying to develop as a drug for human cytomegalovirus. The drug has successfully completed phase 1 of clinical trials, and we're now trying to raise funds to get it into phase 2. So we have a lot of things to do to keep pushing this compound forward. I have been recently involved with several experiments since I joined Microbiotix, among which we were interested to look at synergy with other FDA-approved compounds, such as letermovir, which was approved last year for prophylaxis. And we're looking at other aspects for developing this compound. Recently, we picked up activity against adenovirus as well, TWiV. And we're trying to see whether we can develop MBX400 as an anti-adenovirus as well. And both CMV and adeno are important for immunocompromised patients, so that fits the niche of the compound. And in addition to helping with MBX400, I work on several other discovery projects because we have a wide spectrum of interests. I currently work on a project with an aim of developing entry inhibitors for influenza viruses, since I come with a little bit of experience on flu. So we have a series of compounds that came out of a high-throughput screen using an HIV pseudotyped vector with a luciferase reporter. And we came across some interesting compounds which bind to the HA stem and interfere with fusion. They are very potent against type group 1 HA, and some of the work that I'm currently involved with is trying to expand the potency against group 2 as well. In addition to MBX400 and flu, we recently had some funded grants on Zika. So I'm also trying to push this forward, and we have several other work that you will hear more from Grace about that I'm always happy to help with.
So how do you pick your targets? Let's say CMV. What was the rationale behind that?
Well, that was picked long time before I joined the company. But this is a classic nucleoside inhibitor. So it gets phosphorylated by the UL97, and then the monophosphate is converted to the triphosphate form intracellularly, and then it inhibits the CMV polymerase. So that is the target, a classic herpes virus target. But we have other projects also on CMV trying to find novel targets. We recently ran a screen, and we found some early stage inhibitors, and we're trying to raise some more funds to continue working on this interesting compound.
So MBX400, the screen was designed to find polymerase inhibitors, nucleoside-based ones?
I think Terry will know a lot about this.
Yeah, these were ganciclovir analogs. So yes, they were specifically designed primarily for herpes. They've been tested for spectrum, but they are fairly specific for beta and gamma herpes viruses. And we just picked up, our collaborator Mark Pritchard at UAB just picked up the adeno activity, and we just published on that a couple months ago.
Now the adeno is important because, as you know, we just had a severe outbreak in a children's home in New Jersey. And the physicians said there's nothing we can do, so having a drug would be great.
It'd be great. We agree.
Now for CMV, you said transplant patients who are immunosuppressed. That's a big target, right?
And congenital infection as well. But currently we're focusing on transplant patients.
So by congenital infection, do you mean if a child is born or the mother? So the mother's pregnant and infected, you could treat her.
Right.
So that's a tricky population to develop an antiviral for. Right?
It is, of course. And that's why we're trying to make sure that when we get to that stage, we have a very safe compound for this type of patient.
So you mentioned one licensed anti-CMV compound, right?
No, we have another program.
Another company's, did you mention that?
Letermovir.
Yes.
Letermovir is an FDA,
So there's just the one, there's nothing else.
Ganciclovir is the most commonly used compound, but recently letermovir is a terminase inhibitor, but it was approved for prophylaxis only. And we're hoping that I've actually shown that our MBX400 works in synergy with letermovir, and the current trend in the whole field is developing combinations, drug combinations. So we're hoping that at some point we will be a part of the drug combination that's used for treating HCMV.
So obviously, if you want to have a combination, you need to test them in patients together.
Absolutely.
So if you have one drug and another company has another and a third, how do you get together?
It's a very good question. What do you think, Terry?
Well, you first ask them.
And
and depending on the answer, you may or may not have to supply it yourself, right? So there's both opportunities, but it's much easier to work with them, obviously.
Of course, yeah. And hopefully you cooperate, right?
Hopefully. That's the goal.
Because if you sell the compound, you share it, right?
That's correct.
As well.
Yep, sure.
So you said, now I understand why you're interested in herpes viruses and Alzheimer's disease.
Yes, we are.
Because if it turns out that they're involved, then you may be in a good position.
We could be in a great position.
Yeah. Maybe I forgot to mention that MBX400 is highly potent against beta and gamma herpes viruses. And some of the recent work that showed some real connection, but it's not proved yet. But HHV6 and HHV7 are viruses that MBX400 is highly active against.
Okay, so less so against simplex.
Less so against alpha herpes viruses in general.
Basically inactive or poorly active against the alpha herpes virus.
But I'm sure if we found out in 5 years that herpes simplex type 1 was involved in Alzheimer's, you could get involved in a program for that, right?
Well, developing a combination between acyclovir and MBX400 should cover the entire spectrum of human herpes viruses. So that would be a very good combination.
So you think acyclovir would be okay for
I hope so. Yes. it should be.
simplex.
Yeah, it's one of the safest drugs ever. As far
In
as
fact,
I know.
that's the clinical trial that's enrolling at Columbia now. I think it's valacyclovir
oral form.
for Alzheimer's. It's remarkable.
We do have backup compounds that are active against the alpha viruses
Yeah.
as well.
So the flu project, is that something that was also here before you came?
Yes, there's a paper that was published, I think, in 2014. And currently, we are trying to move this whole project forward by expanding the spectrum of activity. So these compounds inhibit H1N1 with very low IC50. And now we're trying, with help of our chemistry colleagues.
And Mike Caffrey at
And
University
Mike
of
Caffrey.
UAB
at
University of Illinois, Chicago.
University of Illinois, to introduce some changes to this backbone and make it cover the widest spectrum possible of all types of HA, so it becomes a broad spectrum flu inhibitor. And I should mention that it's very active against oseltamivir resistant H1N1. So again, going back to the idea of developing drug combinations.
All right.
And just to add to that, that's a program that grew out of work Mike Caffrey was doing, structurally characterizing the interactions of these compounds with these proteins. And so it's a very much of a structure-based drug design strategy that we're using in this particular instance.
So problem with flu antivirals, there are now a couple on the market, right? And the newest one is an endonuclease inhibitor.
Baloxavir.
Yeah.
Yeah.
You have to give them quickly, otherwise,
Sure
24
do.
to 48 hours. Is yours going to be any different? And if not, how do you deal with that?
Well, in case of a pandemic, as you know, anything will help to reduce mortality, right? So yeah, the quick diagnosis and quick therapy will have a great impact. But I mean, we have examples from the 2009 pandemic. I mean, people who were partially immune via taking the seasonal vaccines had less mortalities. So anything will help. This is a very serious virus.
Especially in an institutional setting, elderly
Absolutely.
people or kids. If an outbreak is beginning, you can go in and dose people.
Right.
Immunocompromised, for example.
Yeah.
That's a big need.
And I think that's something that we will likely at least explore early on in the discovery process, is whether there's a prophylactic strategy available.
Right.
Particularly in an elderly home setting or something along those lines, a hospital setting, where there's a incidence of infection, you might treat the surrounding patients.
So does that require a different kind of trial than a treatment trial?
Yes, and it sometimes requires a very different safety profile, and these are questions that we need to understand before we can really commit to where we're going to go.
Yeah. So you have to dose people without infection, so it has to be safe, and it has to last long enough so that it would protect within so many days, right?
Or the duration of treatment has to last longer.
Okay, got it. Oh, there have been good examples of prophylaxis, right, for HIV, for example. So that can be a good approach. So I'm sure you want to make your own mark and find targets of your own. Is that something you can do? Can you come up with targets and pitch them around this table maybe? And
So yes, of course. Well, I should probably mention that we're lucky that we started working with you.
is that right? Did you?
Yes.
Oh, that's right. You did.
Yes. So we have some interesting group of compounds that came out from another screen, totally irrelevant to what we're currently trying to do, and they turn out to be inhibiting a big group of RNA viruses. And we're hoping that these compounds can be developed as broad-spectrum RNA virus inhibitors. And we're lucky enough that you did some experiments, and we're hoping that we can move this forward to become something more promising and something we can all add our own fingerprint to it.
So in other words, you can be flexible. You can see what's going on. You can see what new viruses are emerging and adapt your programs to that. You're small enough to be able to do that.
Sure.
Absolutely.
And what I really like about my work here is that I'm not stiff to what I should do or what I can do. It's very flexible as long as we can come up with something reasonable. And I really like being able to jump between different projects. I'm very greedy when it comes to viruses. So I don't want to work on a single virus. And I'm lucky that the environment at Microbiotix are helping me to work and learn more about other interesting viruses.
So can you walk into Terry's office anytime and say, hey, I got this idea?
Absolutely. I just did this last week.
Yes.
Happens all the time.
So my wife was at Merck for 27 years. We were eating in a restaurant in New York a couple of weeks ago, and guess who walks in? Roy Vagelos and his wife, who was CEO while she was there. I said, go say hi. And she said, are you kidding? It's different, right?
It is different.
Grace, let's hear a bit about what you're doing.
All right. So here my projects are more about working on small molecule screenings. So like Terry told you, we have a lot of excellent collaborators. So in academia, so they look at viruses and then find druggable targets. Then they develop some experiment to characterize the interaction, how these proteins behave in cells, and then transfer the technology here. And then we have a collection of small molecule libraries. So we screen them and then see which small molecules that can give us the results we want. And then we handed over the small molecules to chemistry, and then they can tell me like, oh no, I don't like this compound, or oh, this one is super good. So
it all depends on the libraries that you start with, right? And you can
Yes.
start with different kinds of libraries depending on your target. Is that right?
Yep. And the collection of small molecules, as I can tell you, makes a big difference in the outcome based on their structure and their chemical properties.
So you said you identify a druggable target. What does that mean?
So one of my projects is on Ebola. For example, when you look at viral replication, you can see that for Ebola, there are 4 viral proteins involved in viral replication. And that includes nuclear protein, VP35, VP30, and protein L, which is a polymerase. And in my previous lab, we identified the VP35 as a critical component for the integrity of the replication complex. Without VP35, there's no replication because nuclear protein and L simply don't interact with each other. And you rely on VP35 to bridge them together. So that way we found out that if you interrupt the interaction between nuclear protein and VP35, you can inhibit viral replication. So from there, we developed an assay to look at the interaction between the 2 components and then transferred the experiment here.
So people have always told me that interrupting protein interfaces is hard.
Yes, it is.
Do you need a big molecule or do small ones work?
So far, we found a small molecule, I think molecular weight around 400, a little before 400. That works pretty potent in our case. So I would say, yes, it is feasible.
Okay.
And I think there's a drug so far approved, only one drug so far approved by FDA, that's small molecule targeting protein-protein interaction.
Is that a virus target or something else? Something else? Okay, so
I would say something
it's
else.
one out of many. So most target enzymatic activities of some kind, right?
That's correct.
Typically.
That's easy. Low-hanging fruit, right?
Yep.
Typically.
So you said Ebola is one of your targets, and I just have to ask you, if it worked out and your chemist made nice active compounds that went to the right places and all that, and you went into people, then how would you would depend on an Ebola outbreak to test the drug, right?
Yes. Well, but
Zach
we
seems
can,
to disagree.
well, not in human, but we can test them in different animal
course.
models. Like one of the closest one would be non-human primates.
Right.
That would be a good representative of human targets. So, and a lot of the vaccines we have right now not on the market, but have been proved that they are active and potent in these animal models. So,
But ultimately, you have to test efficacy in people. And that's always the catch for diseases that are sporadic, like zoonotic diseases, right? I mean, flu is easy because it's X all the time.
There is a recent example of inhibitor of pox virus, smallpox, tecovirimat, which was approved without even testing in humans.
Right.
Because
Of course, we
So
talked
there
about
are-
that a lot, actually, because it's an interesting precedent,
Yeah.
right? But you have to convince the FDA that all your animal data are indicative of what happens
Right,
in people,
right.
right? So you're thinking maybe you could do the same with, is that what you were thinking about? Zach?
they refer to it as the animal rule at the FDA.
Right,
and it's
2 animals, right?
Right, 2 animals as well as clear safety in humans. That said, I believe that approval through the animal rule is considered, I don't want to say temporary, but it's somewhat pending uses in humans. So that if there is an outbreak of Ebola after we have something approved by the animal rule, the FDA is going to monitor efficacy during that outbreak very closely. And if it proves that the compound is not efficacious during that outbreak, I suspect that approval would be revoked at that time.
Interesting. I mean, the pox inhibitors have had some history using in other pox virus, like monkeypox. It's been used, and there was some efficacy. So that's part of why I think that went. Ebola is a tough one because, as you know, there's an outbreak ongoing now, and they have a vaccine, but a lot of people are not getting it because of political issues in the country. And the same would apply to an antiviral, right?
That's true.
And another thing, it's kind of off topic, but
It's okay, we
So-
do that on TWiV.
so it's good to have a vaccine, but
Sure.
as you know, vaccines might be strain specific. So we do have small molecules that target not just Ebola, but also other members of the family,
Right.
like Marburg, Sudan. Yeah.
So we should add that your company doesn't make vaccines, right? You make small
Right, correct.
molecule antivirals
That's correct.
because a vaccine is a whole another animal right there. In fact, I pitched an idea to Islam. I have a great idea for a vaccine. He said, well, we're a small molecule company. You got them trained well.
Okay, I'm glad you said that.
So you leave that to other people.
We do.
Because we don't want to imply that there's no place for vaccines in infectious diseases. It's just
Plenty of space.
plenty of space, and you're just doing the
We're
antiviral
focused
space.
on small molecule drug discovery and development.
What other, is there anything else you're working on you could tell us?
So I also work on Lassa virus.
Lassa.
So that's a different fever virus. And we are not looking at viral replication. In this case, we look at viral entry because it's an early stage in the life cycle. So it's also very effective. If you can stop the viral infection at that early stage, you're at a better place. So.
In fact, most people don't appreciate how much bigger Lassa is in Africa than Ebola.
Yes.
Many thousands, tens of thousands of infections.
Correct.
Gets no press.
Yeah.
That's correct.
And really, we need vaccines and antivirals.
Yeah.
Yes, we do.
And it's good to pay attention to them now rather than wait for outbreak.
Sure.
So.
Which begs the question, of course, who would pay for them? But you don't worry about that, right?
We'll let the WHO
We'll make it-
and the Gates
Yeah.
Foundation, et cetera. So we're optimistic there will be payers.
There should be. There should be. I have to tell you that it was Lassa virus that got me interested in virology. Did you ever read Fever by John Fuller?
Oh,
If not, you should read it. It's a great book about the
Yes.
first Lassa outbreak in Nigeria in the 60s. It was really the first of what we call emerging zoonotic infections. We hadn't seen that before. It's a really interesting story. You didn't read it, right?
no.
Go- it's-
I heard about it.
You should buy it. It's you'll have to get a used book on Amazon, but it's really good. Part of it takes place at Columbia Hospital, which is where I work. So it's very, very cool. So why did you come here, of all places?
So
Small
actually,
company, kind of risky. You don't know what's happening. Funny funding model. Why did you come here?
that's part of true. The company is small. We have only like 26 people. And my former PI, Gaia, Emma Ressinger from Washington University, is a collaborator of Microbiotix. And one of the experiments or assays we're running is something I developed over there. So I know these people before I came.
She was a legacy.
Got it.
What can I tell you?
Actually, I should point out that being small is one of the strongest points about this place, because we can all fit on this table. There are no complicated layers of management. I can go and talk to Terry at any time. So that actually helps a lot. And we genuinely work together. In academia, there's a lot of friction and competition. We're not competing. We work as a team. So being small is actually a plus.
Yeah, I was saying that to be controversial, of course. I understand, but there are pluses and minuses to big and small and in between, of course.
Of course.
Absolutely.
Although my wife worked at Merck for 27 years, and she went to an 8-person biotech, and she said if I had to do it over, I'd go to a small company because you can, like you said, you can talk to everyone,
Yep.
and people will listen. Whereas when the company's big, people are not listening. So my wife used to say the chemists are king at Merck, and if they're not interested in what you're doing, you're sunk.
We consider him to be a prince, actually.
So tell us what you're doing here.
So I do, as you might guess, since I'm running the chemistry department, I have a finger to some extent in almost all projects. I mean, if it's a high throughput screening project, my entire input is looking at the structures. And I think Grace was very kind in saying that I occasionally say things are good. I'm really, unfortunately, fairly negative in that I either say I don't like something or I don't know, let's try it. Those are kind of my two options. The more advanced projects is where my attention tends to focus more intensely, where we get compounds out of these high throughput screens, which have, in many cases, moderate potency, horrific physical properties, and questionable metabolic properties. And so it's the job of the chemistry department to start understanding what aspects of the structure of the molecule dictate those different components of activity, and how to really do the multi-parameter optimization to bring all of that forward to, in most of these cases, a sufficiently drug-like molecule that we can do a proof of concept experiment in animals, and ideally all the way to the point where we have a viable preclinical candidate that can go into serious safety and pharmacological testing. The viral project that I probably am most closely involved with is related to the Lhasa project Grace was talking about, which is actually an Ebola entry program. And that again came out of a high throughput screen of Ebola entry inhibitors that was a collaboration with Kartik Chandran. I hope I'm not pronouncing his name terribly wrong. Apologies, Kartik, if I am. Where we're looking specifically at the viral particles binding to the host, and it is again a protein-protein interaction that we're looking to disrupt. And we found small molecules that do so successfully. We don't yet know exactly from a molecular scale level what the mechanism is, but I'm suspicious that we're having an allosteric effect, that we're changing the conformation of one of the key proteins, and that's disrupting the protein-protein interface. But that is far more of a suspicion than a known fact. The compounds that came out of that screen were actually quite soluble, but they're actually fairly amphiphilic. They're very greasy molecules. And the problem with greasy molecules is they tend to stick to a lot of things. And so the focus in that program has been overall increasing the surface polarity of the molecules as a way to increase specificity and remove off-target effects that are most readily visualizable in the context of cytotoxicity. And so we've been able to get compounds that are quite soluble, they're quite well behaved, highly selective. We have one series where we've been able to actually do fairly high dosing levels in mice without any toxicity effects. I did learn a harsh lesson about efficacy testing in mouse models of Ebola, where we did a model and showed that our vehicle was quite effective in curing Ebola-infected mice, which it turns out, at least in the mouse infection with Ebola, in many cases dehydration is the primary mechanism or cause of death. And so if you give them too large a bolus of water, that can have a very helpful effect on the mice.
Actually, for Ebola, good supportive therapy is very important for survival. As we saw in the U.S. infected, most of them survived. You know, so.
Yep, absolutely.
Which I'll admit, as a chemist, it's a little disheartening to think that I'm trying to improve on water as a drug.
Water's a tough one, yeah. So starting from the beginning, so when they're designing screens and they're looking at libraries, are you participating and saying, no, that wouldn't be a good library, or this one would be better?
At the level of screen design, no. At the level of library acquisition, And whether we're starting to deprioritize libraries, to some extent, yes. And it's actually fascinating. Our library, I think we have about 280,000 compounds currently in-house that we use. And it's subdivided functionally by date of purchase. And it's very consistent. If I get a screening set from a 10-year-old set of compounds, if there are 50 compounds there, I'll probably reject 45 of them. If I get a screening set from a library that we bought 6 months ago, if there are 50 compounds in that set, I'll probably reject 2 of them.
Right.
And and I think that says a lot more about the libraries that are commercially available, that are being generated. It certainly isn't an aging issue that our libraries are old chemicals or anything. It's just that the libraries have become much more drug-like. Part of that is we've also, I'll admit, the older libraries I was not here for the purchase of. I suspect they were the best available at the time. We're very selective now. We take a very good look at the overall predicted physical properties of overall library molecules and are selective on that basis.
So these 280,000 compounds, they've mostly been purchased but you're also accumulating a library of your own modified compounds, right?
So we have about, I think it's 5,000 in-house synthesized compounds that have come out of any given number of programs. Those don't typically go into our high throughput screens at this time. And I think the reason being that we already have a pretty good idea of something they do, and so if we don't want it to be doing that, that's an added problem. We have recently piloted a program actually involving my graduate advisor's lab where we received about 300 synthetic intermediates that had percolated throughout his career of complex molecule synthesis. And they've gone into our screens. It's still very early days on that. The long-term model is that any of those compounds that come out on a hit, they would actually play the medicinal chemistry role on that program moving forward. And that's something that I don't know if we've clearly delineated about our collaborations, is we really have the full set of skills necessary for drug discovery internally. All of our collaborations involve somebody playing one of those components, and we have the parts of the company that are relevant collaborating with them. And so there are programs where the collaborator is a medicinal chemist. And in those programs, my role is primarily to be a second set of medicinal chemistry eyes, as opposed to my doing any optimization or anything along those lines. And so it gives us a lot of flexibility on our collaboration.
The reason I ask is because, again, drawing from my Merck experience, they had hundreds of thousands of in-house synthesized compounds because they had a long history, and they wanted to screen them. And at one point, they got every employee to spend one day a month weighing out compounds and putting them in vials for testing because they weren't aliquoted in that way. And they all got T-shirts and hats, which had some slogan to make them all happy to do this when, in fact, they didn't want to.
Well, and I mean, I think the hard question a lot of pharma companies have been asking themselves is what's come out of that effort? And it hasn't been as successful as they were projecting in the mid to late 90s. All these high throughput screening campaigns were going to be. I think the advantage that we have with our high throughput screens is that it's very mechanistically based. And in many cases, what was the original hit compound, and after a relatively short number of iterations, what is the compound we're still looking at? We've really changed the structure significantly, and it was the lesson of what does it take to interact with this component of the molecule that we learned from the screen, as opposed to what is the key chemical matter.
So let's say you have a hit from a library that's active, and it's a bit cytotoxic, but that Islam or Grace want to pursue it. Can you look at this and say what to change, or is it more of a hit and miss proposition?
There are a few obvious red flags that you might look at and say this is something we want to change. Usually things that boil down to something as simple as log D, so the overall polarity of the molecule, whether it's strongly amphiphilic. Those are things that you typically don't want a highly lipophilic molecule. They stick to everything. particularly, and even worse actually, are cationic lipophilic molecules, which unfortunately I spent my PhD making cationic lipophilic molecules that had amazing biological activity. I can think of one molecule where I think there were 10 or 20 very interesting protein-protein interactions that disrupted, which in hindsight is a terrible sign. And those factors we can take care of quickly. More often what we do is, if Islam comes to me and says, I have this compound from our library, it looks very exciting, the first thing I'll do is a substructure search in our own library and say, okay, test these 20 other compounds, and let's look at that set of data. Because one of the first things you want to look at is if you change the structure over a course of 20 compounds and your potency and your cytotoxicity march hand in hand, you have a problem. Because you're probably seeing two different readouts of the exact same mechanism. And that's, for me at least, very often a reason to run away. Now, once upon a time, working in the 90s in cancer targeting programs, that was not a reason to run away. That's what we were looking for. We wanted cytotoxic compounds that just killed the cancer faster than the host. With virology or antibacterials, we don't want to hurt the host at all. And even in modern oncology, you don't want to do that either.
So in similar ways, you can say modify a compound so it lasts longer in the blood or goes to the right places and so forth. Right. You're beginning to see what kinds of molecule features are associated with favorable properties, right?
Yeah, and so we do that through a screening cascade or assay funnel. Typically, we look at potency, cytotoxicity, and then very quickly we start asking questions about solubility. We have a assay called a microsomal stability assay, which are essentially miniature livers. And we ask how quickly the miniature liver chews up the compound. We do serum stability, which is a question of will the compound survive in serum. And then serum binding, which is a question of whether the compound, as soon as you put it into an animal's bloodstream, will it be completely sequestered or will it be available for activity?
Keiko?
So Keiko-2, it depends on the program. So for a program where we want oral bioavailability, Keiko-2 permeability is critical, and that is Keiko-2 cells are essentially gut epithelial cells, and you're simply looking at whether or not a molecule can move through them successfully, as well as whether any of the PGP efflux pumps or other efflux mechanisms are going to be pushing the compound out.
So we've mentioned animals a few times, but you don't have animals here.
We do not.
So you have to send everything out for animal testing?
Yeah, so either through collaborators or vendors, depending on what we need to be done. What we do internally is we do some of the bioanalytical support. So if it's a PK study, we can do the analysis, for example. Right. So someone else will do the dosing.
Right. So Terry, I see here on the wall there are lots of posters which suggest you've put these at meetings, because you probably don't make the posters just for here, right?
That is true. We go to a lot of meetings.
So you're giving this information at meetings.
Right.
There are publications.
Plenty of them.
On your website. So you encourage the dissemination of your findings?
Yeah, we do, because once again, the model here, because we're very heavy on grants, we have a lot of academic collaborators and government collaborators, and for a lot of reasons, we need to publish our work. Obviously, we file a lot of patents, so we'll have patents in place before the publications or the presentation.
Right. So Lynn Enquist is a well-known herpes virologist who worked at DuPont for many years before going to Princeton. And he always said to me, if you see a paper from a company on a drug, it means they're never going to use it. Is that true?
Partially. Not entirely, but partially, yes. Yes, there is some truth in that. And I think the bigger the company, the more true that is. Okay, let's put it that way.
And I'll admit that there's certainly a bias here. If we have two compounds that look promising, but one of them we have discovered a terrible fatal flaw, that one is, if possible, preferred to be the target of publication. But that's an ideal circumstance, and more often we just make sure we have good patent protection before we publish.
Right. So Terry, you are a small company, which we've heard some of the strengths of, but as you move forward with drugs candidates, phase 2 and phase 3 gets more and more costly, and then if all that works out and you have to market, that's probably more than you can handle. So how do you see going forward you dealing with all that?
Well, first of all, we have no plans to ever market or sell a drug. So even the current drug, which is entering phase 2,
Right.
we hope to show proof of principle in humans, and then we would seek a licensee. We would license it or co-develop it or collaborate. But we are going to remain primarily a drug discovery and I will say early development company. That is our goal. Okay.
So early means phase 2?
Through phase 2, through proof of principle. There's two big hurdles. Proof of principle in animals with good safety, proof of principle in humans with good safety. And we would like to get through those two hurdles and then license and or partner with companies that do have full sales and marketing capabilities.
Okay. And so at some point in the future, currently you're running on mostly grants.
Grants and contracts predominantly.
Right.
That's right.
So at some point in the future, you may have income from products.
It's certainly hopeful and probable. I mean, that is one of the goals, that we would like to eventually have drugs on the market, and we would reap the benefits and put it back into discovery research.
However, if that never happened, and I'm sure it will, but just it's hypothetical if it never happened, you're basically a research institute right now.
In some way.
Running on
in some way.
Contracts and SBIR grants and other mechanisms. Could you continue to do that indefinitely?
I don't know about indefinitely, but we can certainly continue to do it. But the goal here is to do some combination thereof, that we hope to continue to do very good discovery, anti-infective work, which will be worthy of continued funding through various agencies, some of which, just by the nature of the work, you couldn't get VCs to support it. We work on a lot of emerging diseases. We work on a lot of multi-drug resistant diseases. We work on things like tuberculosis and malaria, and those are not necessarily the most popular projects for VCs. So just by the nature of those types of programs, we need alternative sources of funding. So that's number one. But we have other projects that we think will lure venture capitalists and other interested parties as we get proof of concept. So the model is kind of a mix going forward. So,
do you work with BARDA, for example, which is the part of the government that it worked with on the smallpox antivirals, for example?
Yeah, we have talked with BARDA. We've never worked with BARDA, and primarily our projects didn't seem to be quite mature enough for BARDA. However, we now work with CARB-X, and CARB-X was essentially initiated through BARDA. And I look at CARB-X as it's almost the discovery arm of BARDA. So we do have a program that's funded through CARB-X.
Okay. So I'm sure a number of people are listening who are earlier in their careers, and they may be interested in not an academic job, but maybe an industry job or a company like yours. What would you tell them? How should they prepare?
I would prepare just like Grace. I would do an undergraduate program, a graduate program, and an excellent postdoc program in science, in the best lab you can find in an area of interest. And if you do that, everything else will take care of itself. That's my advice.
Okay. Well, you're a unique company. There are not many like you.
That's true. I think we are fairly unique. I mean, there's a few other companies that are more heavy, maybe exclusively, on, non-dilutive funding, as they call it. Right? But very few. And plus, the collaborative model here is, I think, we just have a lot of, I call, high-quality collaborators. I mean, we're collaborating with a lot of really great people, and that has really been, I think, the key to our success. No question about it. So that part of the model in particular is particularly important, and we're always looking for new collaborators.
So let me ask each one of you the same question. What are you going to tell people who are listening who might be interested? What should they do?
I think we talked enough about the unique model on which this company is working. I think before I joined Microbiotix, I was not aware that such model exists. So I think I would love to make this clear to all of our colleagues in the field that this is something. I mean, with drug discovery, academics usually go until a certain stage, and then things become difficult. And this is the space that we can fill in. So I would like to invite anyone who has something that could be interesting to contact us, and we will sit down together and see how can we take this forward. And there is a mutual benefit for both parties, and we do publish, so academics shouldn't be afraid of working with us. So that's what I I would like to make clear to people who are listening.
In fact, you were surprised that you liked it when you first interviewed, right?
I did, yes. And I texted my wife on the way back and I said, this is much, much better than I thought.
I think
Terry hasn't heard this before, but that's true.
I think academics do know, well, we don't know what industry is like.
Yeah.
So we always approach it with a
I was
questioning
very
view.
nervous, and I was lucky that Don, our CSO, met me in the morning, and after a few minutes, I felt at home. And because I have a PhD that involved a lot of work in antivirals, once I started talking to people here, I found that we're speaking the same language, and it was a good fit, I hope.
So far, so good.
Good.
Grace, how about you? What should people be doing if they want to work in a place like this?
Actually, I would say when you look for some opportunities, don't ignore the ones you have already. So I remember in my previous lab, I was working on a lot of projects, and then one of them was the one I did, like on Ebola, that got transferred to here. So if I said no, I don't have enough time to run all the experiments, then I would lose an opportunity to work here. So you never know.
So you had not considered a career in industry, right?
I did.
You did?
I did. Because I think research is interesting, but also I want to see from beginning to the end, from drug discovery, like the target discovery, to small molecule identification, and then to improvement, ICR improvement, and then to later stage of drug development. So I think it's interesting, and you will feel accomplished at some point.
So Zach, when people train as chemists, you had some academics in your career. Is the vast majority of chemists not looking for an academic career, or?
So, I mean, it depends to some extent. My PhD lab was probably 75% industry bound from day one. And part of that is that a total synthesis lab at that time, people said if you want to do medicinal chemistry or process chemistry, you have to do total synthesis as your training. However, my postdoc lab was a lab that was well known for putting people straight into academia. And so in that context, the numbers were almost perfectly reversed. And I'd had an interest in going academic from very early on. And to be bluntly honest, pretty much everything I had hoped to get out of being an academic, specifically real research freedom, the ability to dictate the work I was doing, and it may have to do with my interests aligning, something I find more readily available in industry than I did in academia. In academia, I ran into a problem where in many cases I had to carry water for areas I wasn't expert in. I mentioned that I was doing cellular imaging agents. I was doing the microscopy on that, while at the same time figuring out how to turn a microscope on and trying to get publication-worthy images. Whereas here, if I become interested in a gonorrhea program and I get a piece of data on a different bacteria, I can wander straight into any of my colleagues' offices and say, is this even relevant? And can you put this into context for me? And they're happy to not only educate me, oftentimes they'll say, that's interesting, I'm going to go do this experiment to support and teach you even more. It's a very collaborative effort, and certainly Microbiotix is a little bit unusual in the extent of academic freedom that we have here. But even during my time at Celgene, there was a fairly significant amount of openness to discussing new programs, new projects, or new research directions. I mean, there was always the discussion of whether this was on point for the company. We're not going to spend 3 years researching something that can't possibly go to anything. But in academia, there's a question of grantability that's a very similar discussion. Can you get grant funding for this? And so I find it to be preferable, personally. The one skill I think that people oftentimes underemphasize during their scientific training is communication, both written and verbal, in presentation. I've we've had interview candidates at times, or even cover letters on applications that come across, and I look at it and I think, I'm not sure what research you've performed. I'm not sure what your personal contribution was. I'm not even sure whether this would get a passing grade in a 5th grade grammar analysis. And I think that's a skill that people need to make sure they polish, not just for the application process. It's valuable every step of the way through industry, because it's such a collaborative environment. If Islam doesn't know why I'm asking him a question or what the question means, he can't answer it for me. So.
Yeah, I saw that today.
Yeah.
So in essence, there's no difference in training for an academic career and a career like yours.
I don't think so. We want to recruit the best scientists, right? And as I said, I think, those come through a strong graduate and postdoctoral career. And that sets them up, I think, for either, quite honestly. And I think Zach has also expanded on some of the other qualities we look for. But, it starts with the best scientists we can find,
Right.
Right.
So Terry, what's going to happen in 20 years here? What's your vision?
Yeah, my vision is slightly what your previous question, that I hope the company grows in the sense that we continue to push molecules into development. We're able to partner those programs, and see some return on our hard work, which will continue to fund some earlier programs. I think that's really what I would like to see. So once again, we may never become a fully integrated company, but we'll continue to be a really strong research company that meets various needs around the world regarding, say, emerging pathogens and multi-drug resistance, et cetera. So I would see that we'd probably stick with infectious diseases, even though, like I said, we're already moved on to some parasitic diseases. We have some small projects already. So that's the model. That model, I hope, just continues to grow.
So this is just our second TWiV with an industry. We did MedImmune, big pharma company, and now a small pharma. Right. So is there any other message we should send? Have we said everything, Islam, that we should say?
Well, I have a pick if we're finishing up.
We could do picks. I don't have one, but you could pick one.
Oh, sure. So there's a very nice article published in The Scientist. I think the author, just don't want to, I think the author is Ashley Taylor, and the title of the article is How to Successfully Collaborate with Industry. And I think it encapsulates most of the stuff that we talked about, and it begins with an example about James Allison, who won the Nobel Prize on checkpoint inhibitors, which was a successful example of a collaboration between academia and industry. And it also highlights the most important things that academics need to have in mind before starting a collaboration of this kind. So I would totally recommend it to all of my colleagues to read this interesting article.
Although Allison had trouble selling his idea at first.
Of course,
right?
Yeah.
They said, who cares about curing cancer in mice? But you have to be persistent, I guess.
Yeah.
All right. Well, that is TWiV, a special episode here at Microbiotix. And you can find TWiV on any podcast player. You probably listen on your phone or your tablet. You have a podcast player that you use. Please subscribe to TWiV. Go to the subscribe button and hit it. That way, even if you don't listen to every episode, you'll get it. That helps us with our numbers. The more subscribers we have, the more funding we can get and do more things that help communicate science. And if questions and comments, always twiv@microbe.tv. And if you really like what we do, consider supporting us financially. You can go to microbe.tv/contribute. We have a number of ways that you can do that, including a Patreon contribution. My guests today have been at Microbiotix. Where did that name come from?
That was one of our founders, Neil Brown. So that's
came up with it?
Came up with a combination of microbiology focus and the biotech industry.
Not bad.
That's where that came.
And you didn't have to pay for it.
That was free. Neil Brown, one of our founders. Yeah.
My guests have been CEO Terry Boland. Thanks for joining us.
You're welcome. Thank you.
Director of Chemistry, Prince Zach Aaron. Thank you very much.
Thank you.
Senior Scientist Grace Liu, thank you so much.
Thank you for having me.
And Senior Scientist and my old friend Islam Hussein, who has offered his house and hospitality the last few days.
It was our pleasure.
So thank you for arranging this in yesterday's TWiV and for taking care of me. I appreciate it.
Please come back again.
I will. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM for their support and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.